Alright, I think it's time to start. Hi everybody. This is the last full week of class. How are we doing? Okay, so good news and bad news. Good news, this week kind of at the payoff of what we've been working for this entire semester, like, we're gonna make proteins. Also, it's the last week, so it's like you're being attacked by millions of small, I don't know, ducks, right? Like, this paper's due, that paper's due, all the things. You're doing great. Doing great. This time, next week, you'll still be attacked by ducks, but this time, next week, plus 3 days, you'll be chilling, playing with your dog, watching dumb TV shows, sleeping, whatever it is. Just, you got this. Okay, today's class is normal, Wednesday's class is normal. Friday is, in-class a review of, like, the entire semester. Oh, Monday, you have your exam 3. That's going to actually open early. It's going to open on Friday, so if you want to take it Friday, Saturday, Sunday, or Monday, that's fine. But it closes Monday night. Yeah? This week you have lab exams, so instead of going into lab, you're going to take your lab exam online. You can do it during your lab time. You can do it anytime after 9 tomorrow. I think you'll do great. I'm currently writing it. I think you'll be fine. Yeah, I will send you, like, actually better emails because apparently I've been confusing people. I'll send you so many emails this week, be like, hey, this is the thing to do. Hey, this is the thing to do. Hi again, I feel like I'm not spamming your email enough. I know, right? Yeah, good. Okay, so, this is where it gets good. I mean, I've been saying this the whole time. This is where it goes from gene to protein. Alright? So a couple things. The phenotype of an organism, right? The characteristics you actually can see. Whether that's blood type, whether that's speed of carbohydrate digestion, whether that's the specific type of proteins in your toenails, whether that's slime mold color, right? Whatever it is, those things you can look at, most of those are controlled by proteins. Right, so cells have all sorts of stuff in them, right? They've got lipids, they've got nucleic acids, they've got water, they have salt. But proteins are going to drive the whole thing, right? Because proteins are going to be enzymes. So you maybe have a protein for lipid digestion, right? Or you've got a protein to make bile salts to help digest lipids. So the proteins are going to influence basically everything. And you have so much protein, like tons. And it's not just you, sorry, it's all life as we know it. Whether that's bacteria, or mold, or moss, or lichens, or the dinosaurs, or whatever. Right? So, going from, the pieces all come together with the DNA, that's that heritable information, that gets passed between organisms throughout generations. But that heritable piece is then read to build the proteins. Yeah? Gene expression, which is the idea of reading the DNA to actually mean something, that's not done in one straight shot. There's a 2-jump process. The first part is something called transcription. Has anybody ever transcribed something before? You all seen old-timey movies, right, where, the old-timey secretary is typing as somebody's dictating. Right, someone's like, "Dear sir, blah 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 blah," and the secretary's blah 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 blah. Right? Or maybe, I don't know, when you're writing a paper, you're reading a book and you're like, "Oh, I need the information that's in this book. I want that information onto my Word document." Right? this has happened. Or actually, best example we're gonna go for you're typing up your notes. Some people do this, some people don't, most people don't. But if you're the person, you've got these notes, and you're going to go from your handwriting on the page to typed up on the computer. Okay? Your notes on the page, what language are they in when you write them? English? Okay, cool. Yeah, that makes sense. Like, go you. When you type them up on the computer, what language are they in then? Still English. Okay. Why? Not why do you speak English, yay for speaking English, but if you had the notes in English and you're now putting them in English, what? Why bother? They were in English a minute ago. Just because you feel like, right? Why not? Right? But are you okay that? Sorry, keep going. You're on it. You just want them in a different form. Is that fair? But you're still cool with English. Is everyone okay with this? Right? Transcription does not change the language. So transcription, when you're doing it in anything, transcription doesn't change the language. Someone's telling you things, you're transcribing it out for them. That's not changing the language they're speaking. It's just changing it into a more usable form. Maybe you have this whole big book of information, right? You don't want to lug the entire book around. You open up the page that you want. You don't tear the page out unless you're a horrible human being. Not really, sorry, but don't tear pages out of books. You don't tear the page out, but you take notes from it, right? You transcribe from what you have into the same language, but in a more usable form. Fair enough? So transcription in biology is the same deal. You're going to read DNA. The language is nucleic acids. DNA is in nucleic acid. That's what the stuff's made of. Right? So you're reading DNA. You're going to, when you transcribe DNA, you're reading DNA and you're going to build RNA. RNA is still nucleic acids. They're slightly different nucleic acids. The sugar has one more oxygen on it, but it's the same stuff, except for the oxygen and sugar. But like, it's in the same language. It's in the language of nucleic acid. Okay? So now you've gone from your DNA, which when you're transcribing, you don't destroy the original, right? So you've got this book that you're taking notes from. As you take notes on someplace else, you're transcribing some of this. That does not destroy the original book. You just now have the information from the original book in your new book or your new notepad. Fair enough? You transcribe information. So transcription, still nucleic acids going from DNA to you make RNA. Fair enough? Concerns about this? That's transcription. Has somebody ever translated something? Yeah? What language? I'm curious. Spanish. Going from Spanish to English or English to Spanish? Either. Okay. We're okay with this idea? So, do you mind if I just like pick on you now? You're like, sure, get used to it. You're, I don't know, what do you want to translate? You've got some idea about like, I think of a good example. I don't know if I have a good example. You're wanting to buy guinea pigs for your little brother for Christmas. He would love them. They're great. Your parents are convinced by this being a good idea, but you're like, yes, I'm going to get a male guinea pig and a female guinea pig. He can have all the guinea pigs. Right? Okay. So if you have that idea, that concept in English, right? And you're going to translate that concept to your grandma. Because you can go ask your grandma because your parents, they're not buying in, but your grandma might buy into this. If you're translating that guinea pig idea to your grandma, you keep the same concept. The guinea pig stays. Yeah, we need to do this is awesome, that stays. Yeah. What changes? Just the language? Yeah. Fair enough? Are people okay with this? But the language totally changes. Right? Does anybody have a hard time with the concept of translation? You go from one language to the other, but you're trying to have the meaning come through? Yes? Okay. translation, you're going to go from the language of nucleic acids, right? You're going from RNA, which is in nucleic acids. You're going from that, and you're going to make a protein, which is made out of amino acids. It's made out of an entirely different stuff. But the information on the RNA was coding for a specific type of amino acid. So that information is now represented in amino acids. Is everyone okay with this? This slide's just lots of words to say this, which is, okay, extended analogies besides, what's happening is, alright, in like you're looking in a bacteria or something, you have DNA. It's great. During transcription, that DNA is read, it's not destroyed, but you read it, and you use it to make RNA. In this case, well, use it to make messenger RNA. Okay? You're not expecting to read the whole entire, all the DNA that's there. You just want some of it, right? You're reading this book, you don't want to handwrite the entire thing out, you just want a quote from it, right? So transcription happens, you go from DNA, you make mRNA, then from there, your helpful ribosome can come along. The ribosome can read the mRNA, read something that's in nucleic acid, and use that nucleic acid information to build a series of peptides to build a protein, right? So from here to here is transcription, from here to here is translation. Yeah? I realize August was like a lifetime ago, but prokaryotic cells, like bacteria, do they have nuclei? Do they have a nucleus? Nope, no nucleus. So they don't have a nucleus. So what can happen is, as this mRNA is actually being made, is being transcribed from DNA, the ribosome can come along and start going for it, can start translating it into a protein. Eukaryotic cells have the same basic idea, but there's a nucleus. So transcription happens like normal. You go from DNA to mRNA. In eukaryotic cells, you actually like shine up the RNA a little bit. You process it a bit so it can leave the nucleus. But then mRNA is pushed out of the nucleus. So transcriptions happen. You push the mRNA out, and then translation happens. Your ribosomes come along and translate information here into a polypeptide, which will be a protein. So far, so good? Questions? We're doing okay. This picture, some version of this picture, I'm going to have on the final. Like, it will be on the final. But you all know this, right? You've got information on DNA. You read that information to make RNA. This step is called what? Translation or transcription? You so know. I know you know. Yeah, it's transcription because you're still nucleic acid, right? So again, this is not destroyed, but you're reading this to make this. Then you read RNA to make a protein. This step is called translation, and it goes in that order. We need to take a brief aside and do some stuff on the board. And I'm going to try very hard to have it work for the online people. Because I got better chalk. That's bad. Gonna break my neck one of these days. Okay. Nucleic acids. Remind me what letters I get to play with for nucleic acids? A, G, C, T. Everyone's cool with this? Okay. Sorry, I'm trying to make sure I'm on camera. So I've got A, G, C, T. Cool. I need that. I need those individual nucleic acid bits to code for amino acids. Okay? Because I need to be able to read this and be like, oh, amino acid whatever, or like tryptophan, right? Okay? So the absolute most straightforward way to do this would be like, what? I'll just have one nucleic acid code for one amino acid, right? So it'd be like, well, let's just say T equals amino acid number 1, okay? I'm going to keep amino acids in white and nucleic acids in green, okay? And then maybe A codes for amino acid number 2, and G codes for amino acid number 3, and the only one I'm missing here is C codes for amino acid number 4. Fair enough? Problem with this, are there more than 4 amino acids you use to build proteins? Yes. How many amino acids are there to build proteins? 20, which is bigger than 4. What do I do? If I want codes that code for 20 different amino acids, but they only have those 4 letters, is it even possible? I'm going to need more than one to code, yeah? Okay, so what happens, we're going to erase these, we're going to try something, we're going to try with 2. What happens if I'm okay with having 2 2 nucleic acids in every single code? Like this idea? So if we've got 2 amino acids, help me out with what we have. We have something like AA is a unique combination. What's another one we could have? Let's go along the list. How about AG, AC, AT? Those are all unique codes, right? They're all unique from each other. I could have GA, GG, GC, GT. But wait, there's more. I could also have CA, CG, CC, CT, and T's last one here. T. Okay, those are all unique, right? They're all 2 nucleic acids long now. How many different amino acids could I code for there? Hold the thought first. With 1 long, I could code for 4. With 2 long, how many can I code for now? How many is that? 16, yeah. So with going from 1 to 2, I got a whole bunch more, right? So I can code for a total of 16. Is that enough? I need 20, right? If I have a 3 code chunk, I can actually make 64 different combinations, or 64 different unique little codes here, which is way more than I need, right? I don't need 64. But I have to have at least 20. So having a code that's 3 long is the shortest code where you can get every single amino acid accounted for without having any guessing. Are you okay with this? And that's actually how the system works. You're going to use what's called a triplet code. And the triplet just means there are 3 nucleic acids in it. So, it looks like this. And we'll come back to this slide just a second. looks like this, where you have your DNA. It's double-stranded. It's beautiful. You read the template strand because you want the information that's right here. You want the information that's in the non-template strand to be used to make the protein. So you read this template strand, and DNA polymerase makes DNA, RNA polymerase makes RNA. So you have RNA polymerase come along and read this and put the complementary base down to make mRNA. One quick note, aside from the going from double-stranded to single-strand, any major difference we can see between that DNA strand up there and the mRNA strand down there? Anything that's missing? Do we still have any A's? G's? C's? T's? No, the T's gone. What's instead of a T? U. Okay, so uracil replaces thymine, so you have a U instead of a T in RNA. So in DNA you've got A, G, C, T. In RNA you have A, G, C, and U instead of T. Alright? Uracil and thymine are really, really similar. They're really similar, they're just like slightly different versions of the same thing. Really similar, but you'll see them there. So you have 4 different options you can use up in DNA. You still have 4 different options you can use for RNA. You just have replaced the T with a U. Okay, is everyone fine with that? Great. Then, so transcription has taken it from DNA to RNA. Then you're going to read that information, and you're going to read the triplet code, and you're going to make the protein from that triplet code. So this set of 3 nucleic acids codes for tryptophan. This right here, codes, it says, when you see UGG, that means tryptophan. So when the cell is building this, it sees that code, like, oh, I know which of the 20 amino acids to grab and attach here. Yeah? We're coming back to that. You have a template strand. The template strand is the one that's red, but then you make the complementary strand is the one that's actually the mRNA. The piece of mRNA is read in the same direction. It's read in the 5' to 3' direction. And the code looks like this. So I'm going to show you the code, and then we're going to talk about all these words. A lot going on here. The chart's actually not terrible. What the chart? This one's saying in mRNA, you could have one in DNA. The only difference would be the T's and the U's. You're saying, okay, got a codon. Cool. All the possible options for the sets of 3 nucleotides are shown here. Right? You can have, where is it? You can have AAA, you can have AAC, AAG, AAU, right? You have all the possibilities, 64 possibilities. It's great. How you read this is, this line right here is the first base. So you have a G as the first base. You're down this chunk. That series up there is what you have for the second base. So let's say you have a G as the first base. You have a G as the second base. This line right here is what you have as the third base. So I want G, 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 I'm right here. See that? These are all the possible options. Great. You'll notice a couple things. First, 64 is a larger number than 20. Yeah? Which means some of these are redundant. Meaning, GGU, GGC, GGA, and GGG all code for the same thing. Right? Also, just make sure we're all reading this, okay? This is the codon. This is the type of amino acid it represents. Alright? So, UGA, that codon, this series of nucleic acids represents tryptophan, the amino acid. Right? Or over here, any of these codons, any of these three nucleic acid chunks represent leucine, the amino acid. Okay? Good? So this code, it's redundant. That means more than one amino acid, more than one codon can code for the same amino acid. This is actually really, really helpful for a number of reasons, but one of them is if you have a mutation in your DNA that accidentally makes it so that instead of being what it should have originally been was GGU and mRNA, it switched the base and made it GGC. That mutation has absolutely zero impact on anything because what it's coding for has not changed at all. You see that? It's great. You can make some errors and still be totally fine. So like, for like threonine or something, so long as you have AC at the first part, the third spot in that codon does not matter. Okay? So it's redundant. There's some like built-in safety. It's not, however, ambiguous, which means every single codon only codes for one amino acid. Sorry, yeah. A given amino acid can be coded for a bunch of different things, but if you have a codon, each codon only codes for one thing. So this GGU codon only codes for this. It doesn't code for any other amino acid. Do you see that? So you can read the code and you can know exactly what to put on. Any combination of three nucleotides, you can read those and be like, yep, this is the amino acid I put on. Fair enough? You'll notice there are a couple other things going on too. There's this over here, AUG. It's the start signal. It's like go. Alright? And there also are three stop signals that once the ribosome gets to that in the mRNA, it means done, we're done, finish it up. Alright? Concerns there? Something to note. Back to the board. I'm pretty sure most of your mothers or grandmas or whomever it is that is the caring adult in your life, you're an adult, but the caring person who's an adult when you were a child, they think you should text them more often. Just so you know. You probably should text them more often. Right? Sorry. It's kind of shot in the dark, but it's like always true. Anyways, we're gonna briefly write a text to them, okay? Okay. We're gonna write, are we gonna write to mom? Sorry, I'm trying to make sure online people can see this, okay? Okay. So far so good? We're okay with that as something to read? It's maybe not the best text, but like, hey mom, how are you? Fair enough? She'd get this? What she's gonna do when she gets this, she's like, oh, yay, I have to do well, whatever, right? She's gonna read 1 word, 2 word, 3 word, 4 word, 5 word, in that order, right? Okay. If there's some slight change internally here that you accidentally mistype and you type it like this, she might still figure out what's going on, right? Still means the same thing, right? There might be some slight changes that if you make some slight change and you completely mess it up, it makes less sense. That's a problem. What's really going to mess it up isn't internal one-letter changes. What's going to completely throw things off is if you get the reading frame wrong. What this means is, if instead of reading 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, somehow, I don't know how this is happening, you start slightly off, and you start here. Then you have words like this, right? Because you've read here. That does not make sense, right? You might accidentally end up with some things that are real words, but it is garbage. Do you see this? Okay. So, you have to read these in what's called the correct reading frame. Because every single combination of 3 nucleotides means something, if you're off, you just have it mean a whole bunch of random junk. Okay? You don't want that. Light. So far, so good. We have this code. It's beautiful. Means a whole bunch of different things. It's this triple code that codes using nucleic acids codes for which amino acids to put to build your protein. Great. So this code works for you. This is the code that your cells use, right? So they see a piece of RNA, they see a piece of mRNA, and they're like, oh, CUU. I know what that means. Every time I see CUU, I know to put a leucine there. Fair enough? So this works in humans. Incidentally, this is the exact same code in raccoons. I know, right? So that means if there's a raccoon, and the raccoon has a piece of mRNA that says, oh, I don't know, GCU, it means alanine for that raccoon, just like it means alanine for you. Kind of cool. Anything special about raccoons? I mean, besides they're wonderful, but not really, right? But raccoons are pretty similar to humans in this idea that we're both mammals, right? If you're in, what's an animal that's less mammally? A platypus. They're amazing. For so many reasons. I have a whole thing about platypuses. Anyways, it's kind of like they didn't read the manual exactly. They're like, I'm supposed to have fur, but maybe legs. Venom? Can I do venom? Anyways, they do. Anyways, sorry. Stay focused. Not really, but try and stay focused. And our little platypus friends, they have the same code. So if they have something that says GCU on their mRNA, it still means alanine for them too. So it works in humans, and raccoons, and platypuses, and mold, and coral, and mushrooms, and trees, and bacteria, and everything. This code is universal, okay? Which, feel a sense of like, oh, life. Okay, are we all feeling a sense of life? Great. Also, we should say global because we don't know about life other places, but as far as we know, every single organism uses the same code, which has some interesting side effects. Well, not side effects, interesting properties that happen. This code means stuff, right? All it says is when there's a piece of mRNA that says this, put this amino acid there, right? Um, the the same language is being read by all life forms that we know, which does mean something like this. Um, Jellyfish make this glow in the dark protein. It's cool. Have you ever seen jellyfish like this? They're beautiful. It's called green fluorescent protein. If you take the the the, the mRNA, or if you take the gene for green fluorescent protein, right? The series of nucleic acids that say this is how to build green fluorescent protein. If you take that and you put it in bacteria, even though this gene came from someplace else, it's using the same code. So bacteria know how to read that code. Alright? So if it reads that code, it makes the protein that glows in the dark. If you put that same gene, and it's not hard to do, like you were able to cut bits of your DNA out just last week, right? If you put that same gene that glows in the dark into tobacco plants, they glow. Because they can read the same information, right? If you put that same gene into mice, they could glow. See this? Anybody feeling a little bit squidgy about this? No, people are like, that's cool, whatever. Okay, some people are like, I'm concerned about you biologists because I watched Jurassic Park and I'm now freaked out, right? Also, Jurassic Park, everybody's criminally irresponsible, but that's okay. There are some interesting side effects with this, right? So, story time first. There's some ethical concerns around this, and rightfully so. Um, but like most interesting things, it gets kind of complicated. So, like, I had a professor, good guy, a geneticist, and you've heard about how um, breastfeeding babies is really good for them, right? And like formula's good, formula's great, and what the people who are making formula are trying to do is they're trying to make the proteins that you get from like cow milk or soybeans, whatever. They're trying to make that protein very, very similar to the proteins that human mothers make to feed their babies, right? The protein that soybeans make is really similar to the proteins that human mothers make. It's a little bit different, but it's very similar. Fair enough? So the chemists are kind of like poking around the soybean protein to make it more like the kind mom makes. Great? So far so good? Okay. What my prof wanted to do um, was he's like, okay, I know the gene that makes this protein. Right? I know the human gene that makes it. It's like AGG, whatever, right? I know that code. So what I can do is I can take that code and I can tell soybeans to make that exact code. Right? I can be like, hey, soybean, you're making AAAATGC. I want you to make AAAAGCG. Right? Just I want you to make a slightly different version of the protein that you're already making. Right? Or but I put this code in because you can read the universal code because you're a soybean. Everything reads the same universal code. Right? So my poor prof, he's all happy about this, right? He's like, okay, I've got this technology, I can do this, I can make these soybeans grow up this protein, it'll be fantastic. We'll put it in baby formula. The babies don't get sick as much because it's exactly the kind they want. Everything's happy. I'm a hero. Life is great, right? Guess what the U.S. government said about this? No, right? And you can actually understand where they're coming from. Because the U.S. government's like, hold on. Tell us again very slowly. You want to take human DNA, and put it into soybeans, and make Frankenstein soybean, part human, part soybean plants that kill us all. He's like, no, 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 like, I'm not like trying to grow like limbs on this thing. It's just one protein, right? But can we see the concern there? Yeah. Um. So yeah. Thoughts, concerns, comments? Briefly, just to make sure we're all feeling clear out, or at least solid understanding of things. Genetically modified organisms are going to be organisms that you've done something to their genetic code. This does not mean it has to be like Spider-Man, right? Usually it's less exciting than that. Being a mouse takes a lot of things to do to be a mouse, right? One given protein or the other given protein, the basic proteins are pretty much the same, right? Between, say, mouse and people, right? It's going to be like, make proteins for fur or hair, make proteins for eyeballs, make proteins for skin, make proteins for liver, make proteins to digest stuff, right? Like, the actual proteins are going to be fairly similar or even identical, but the how that's implemented is slightly different. Are we okay with this? Um, the the arguments for genetic modification are basically like, people have been doing this for forever. Kind of on accident. So like back in BC-ish times, people would do things like where you have one olive tree that's got like a fantastic root system. This is a really strong olive tree. This olive tree just like, you can't kill it. It's great. Yay, olive tree. But the olives it makes are kind of nasty. Just suboptimal. And you've got this other olive tree, that's a different type of olive tree, that has like puny little roots that don't work very well. But the olives are like, mwah, just delicious. Right? I know, stay with me here. I'm trying. Um, and what people do, and they legitimately do it, they like take branches off olive tree that has tasty fruit, and they make a cut in tree that's strong but has kind of gross fruit, and they put that branch into the other one. It's called grafting. And the, the original tree can totally grow that branch that you just added onto it, right? Because the genetic code for that tree is the same genetic code as this one. They might have different information in them, but to read the stuff that's in the new branch, it's not a problem to read it. Does that make sense? Probably the poster child of genetic modified organisms are super helpful is something called golden rice. Heard about golden rice? Oh, this stuff's amazing. Um, a lot of the world's population has like rice as what we eat so we don't starve kind of thing, right? Like rice is great. It's like rice and whatever else, right? Rice is great. It's reasonably straightforward to grow. You can grow enough of it to support your family. The problem is rice is not like the most nutrient dense food on the planet, right? It's not a whole lot of vitamins. So there's a genetically modified version of rice that has the ability to make, vitamin A. So like, you know how like sweet potatoes or carrots are great for you because you're like, hey, it's got vitamins in it. They basically took that little bit and they're making the rice make vitamin A too. Which is then like, this stuff is magical, right? But then there's also the concern of like, hold on, you're telling me you're just like rearranging stuff, and biologists are like, yeah, and the rest of society's like, be careful. And there are good reasons to be careful. My favorite one that like I have not been asked to contribute on at all for this, but it's being pitched to genetically modify mosquitoes. Have people heard about this? Oh, okay, so uber simplification here. But the idea is like, mosquitoes suck. And I was like, yeah, okay. And they're like, mosquitoes spread malaria. Malaria is bad. I'm like, yeah, okay. Wicked there. Insecticides are kind of scary because like, take a whole bunch of things out, and they can give cancer and like all the things, right? It's like, so, the genius plan, and I am not making this up. The genius plan is to genetically modify a bunch of mosquitoes You've heard this. Yeah. So they're sterile, right? So they can't have baby mosquitoes. And you genetically, stay with me, it's good. You genetically modify a whole bunch of mosquitoes. You've got like Musquelian sterile mosquitoes. And you release these sterile mosquitoes into the wild. And you're like, go mosquitoes. And the idea is mosquito who's sterile, mosquito who's not sterile, who try to mate, all the mosquitoes feel like they're happy and satisfied. But no baby mosquitoes. It's genius, right? That's the plan. And yeah, you see, some people around here have concerns about this. They're having concerns about like, you're gonna destroy the ecosystem. And then people are like, but mosquitoes. Anyways, so are we all okay with just at least the biology behind the things to be aware of? And yeah, the gamut runs from this is amazing, this is making some rice that saves thousands of lives to you built Jurassic Park, that's bad, right? Um, but the science behind it is, hey look, that code, everybody can read that code. Like, any organism can read that exact same code. Yeah? So far, so good? Alright. That's the interesting part, class. There's one more, like, just boring bit to show you today. I mean, it's nice, but, like, it's not gonna, like, make you wonder about, scientists again. Um, when you're making this RNA stuff, right, you're transcribing DNA, you need to do what you probably already know you need to do. You need to start it, grow it, and then end it. Tons and tons and tons of words to say this. DNA is big. You don't want to transcribe all DNA at the same time, right? You don't want to, you can't. First of all, you can't, second of all, you don't want to. So what happens is, before the part that's actually coding for whatever, you have the gene that's actually coding for the protein you want. Before that portion, you have what's called a promoter. And a promoter is basically a ginormous flag in the DNA saying, hey, there's a gene right there. It's important. Okay? So that's a promoter. What are called transcription factors can stick to the promoter. And those transcription factors are going to help this thing that's called RNA polymerase stick. DNA polymerase polymerized DNA. It made DNA. Anyone want to take a stab at what RNA polymerase does? Yep, we can say the same thing? Yeah, this stuff makes RNA. And it looks really similar to DNA polymerase because it's like, oh, okay. I'm going to read the template and put the corresponding um, nucleic acids in a sequence that corresponds with the template I'm reading. Yeah? So yeah, you're gonna have this promoter that's gonna be like, hey, there's important genetic material right there. Transcription factors that help this thing go, and then the RNA polymerase is actually going to go for it. Fair enough? Then what this does, it looks really similar to this whole DNA replication thing. RNA polymerase is going to move along DNA. It untwists as it goes, and it just slowly chugs along. I mean, okay, slowly, like 40 nucleotides per second. So here we have this thing. It's whereas DNA, when it's replicating, it's like unwinding the whole thing, making two. When you're transcribing, you don't need to take apart the DNA. You don't want to take apart the DNA. You just want to like open up this little hole, have RNA polymerase add these RNA nucleotides in order and just keep chugging along. And you're, again, still using the free 3' end to add a new bit, free 3' end, add a new bit that matches here. Do we see that idea? Just kind of pulling it open, adding some stuff, pulling it open. Concerns about this? Nah, how are we on time? We're on time. Okay, that's where we end. We'll come back to this exciting bit on Wednesday.